My name is Amanda Murphy. I'm the CFO of MaxCyte. Our CEO is Doug Derfler. He is not here today in person, but here in spirit. Really happy to be here to share our story, and honored to share the stage with such esteemed companies in the space, as you all are aware. We have many MaxCyte folks here today. Basically, if you tell us to come to San Diego and have good food and drinks, we'll all show up. So we've got our head of sales and marketing, product development, many application scientists, and Sarah Meeks, our head of alliance management. I'm sure you know her very well. Just as an introduction to myself, I actually was an analyst before and covered a number of our partners, including Allogene and several others that have shared the stage over the course of the week here. That's how I got to know MaxCyte, just having obviously seen MaxCyte in all the labs of the companies that I covered. Again, honored to be sharing the stage with such esteemed companies. We did just complete our NASDAQ IPO this summer, so I do have to show this slide. You've all seen it before, so I won't spend too much time on that. Just to give a quick introduction to MaxCyte, we are based in Gaithersburg. We have about coming up on 80 employees and rapidly growing. We develop and manufacture technologies for non-viral delivery via our proprietary flow electroporation system that's been in development for over 20 years. Doug was a co-founder of the company and still is the CEO. We have over 400 installed systems now. We really take a partnership approach with our customers, and so that's what I wanted to focus on today in the presentation. We really do take the partnership seriously. We've got 14 cell therapy developers that are now partners of ours, in addition to many other customers that we sell our instruments to. It's really been just awe-inspiring to see the clinical success of our partners over time, just as the space has evolved in terms of next-generation cell therapy development, with a number of players coming into pivotal trials, as you are aware, which is very encouraging and awe-inspiring to see. I think this is a slide that everyone uses at this point, so I won't belabor the point, but I think it's pretty clear that there's been quite a lot of investment in the space and just almost amazing innovation in terms of next-generation approaches, post the introduction and approval of Kymriah and Yescarta, in terms of trying to improve the efficacy of those drugs, or move into new indications and reduce the toxicity. I think what's been interesting for me anyway, as a former analyst being at this meeting, is just kind of reiterating something that Allison said about the complexity of the biology in this space, and just the real focus on the enabling technology providers like MaxCyte on really providing support and collaborating with the companies in the space that are working to develop the better medicines that we're seeing move through the clinic. I think the other thing that's really interesting is just that the technology providers are also recognizing that we need to partner together and collaborate with each other in order to help address the challenges that we've all talked about many times during this meeting, again, to further help what's becoming an exciting space in terms of solving unmet medical needs. What's also been just absolutely fascinating to watch is the pace of innovation and acceptance of some of the newer technologies like CRISPR. Obviously, we all know that breakthroughs of science do take a long time ultimately to develop, and a number of people involved in those contributions over time. But CRISPR has been interesting. It really wasn't that long ago where we had Dwayne "The Rock" Johnson in a movie where CRISPR gone wild turned into animals that were trying to tear down Chicago. That wasn't that long ago where that was something that people weren't taking all that seriously. And it wasn't that long ago when I covered the stocks that a biohacker could take down a stock 20% because people were afraid that it was a commodity, and as everyone knows in this space that's using CRISPR, it's certainly not easy. It's pretty complex and hard to bring to the clinic and industrialize. And now we've moved so quickly to some of the key contributors to the evolution of CRISPR, receiving Nobel prizes and then human data with CRISPR in 2019. So just the pace has been obviously amazing to watch. So we've now got 400 engineered cell therapies in the clinic. Obviously there's other ways of engineering that have been developed for years, zinc fingers, talons, et cetera. And so the complexity of the cell therapies has grown clearly. From a MaxCyte perspective, this is just our history, and I think the key takeaway of this slide is just that we've been around a long time, over 20 years, and we've spent that 20 years really optimizing our delivery platform. And so it's built to be used in the clinic. It's been optimized based on all of our experience with various different cell engineering approaches, cell types, and cargos. And that's really led to us having several partners, as I mentioned. And I'll get into a little more detail, but again, we've maximized this platform so that you can scale up from the low scale all the way up to clinic, so from concept to clinic. And you can deliver almost any molecule into any cell. In terms of the four key areas that I would think about, or we think about in terms of differentiation for MaxCyte, it's really four things. One, it's performance. So again, being able to deliver any molecules, so that's whether that be CRISPR, RNPs, proteins, DNA, RNA, et cetera, into cells at high efficiency and viability. So ultimately, enabling, sort of facilitating the complexity that's occurring in the space in terms of delivery. The second thing is flexibility. So we have, given our experience in the space over the years, we've been able to sort of optimize protocols. So balancing the cargo that's needed to be delivered, as well as the sort of cell efficiency and viability to ultimately maximize the therapeutic index of the therapies. The third thing is scale. So that's been something that's been a large focus for MaxCyte and a big differentiator. So we can scale all the way up from 75,000 cells in a few minutes of transfection, all the way to 20 billion cells with our GMP compliant platform, the GTX, in 30 minutes. So that's, again, I think enabled us to really have a lot of success in supporting our partners in the clinic, especially with the complexity of the cell therapies that are moving forward. The last point I would say is quality. So we, again, have GMP compatible systems. Our disposables are all single-use, sterile. Our system is a closed system, albeit obviously there's opportunity for ongoing integration across the workflow for clinical cell therapy manufacturing. And it's ISO certified. So those are the four key differentiators that I think have led to our success in sort of partnering and supporting the space. In terms of the challenges, these are well discussed, so I won't get into them in too much detail, but I think the complexity piece is important in that obviously viral vectors, the challenges in terms of getting access to viral vectors and the capacity limitations are well known, and the multiplex editing that's been talked about earlier on, even in this presentation. We have partners who are in the clinic with therapies that have as many as 3 edits. That's really lent itself to adoption of other delivery approaches like MaxCyte with our non-viral approach. Also, regulatory risk, which clearly is a big focus of the meeting. We have a master file with the FDA that now has been referenced over 35 times, so giving our platform that clinical support and validation. And then just vein to vein time, obviously the goal is to get better medicines to patients more quickly, and so the fact that we can transfect so many cells in such a short period of time has been something that's a driver of our ability to partner as well. So just kind of this is our proudest slide, so to speak, and supports, I think, some of the things I've talked about in terms of differentiation and really our support of our customers from a regulatory and field scientist perspective. Some of these names are well known. CRISPR we've worked with for many years. Sana, we just signed them up as a partner. So we have 14 partners now, obviously many other customers, but these cover 75 programs, and of those 75, 15 are in the clinic. So in addition to our master file, we have very diverse programs that are actually in the clinic now on the MaxCyte platform. And we continue to have interest in developers looking to partner with us. And Sarah Meeks in the audience here is our alliance manager, so I'm sure she'll be happy to talk further on that. In terms of the benefit of the partnerships, again, it goes beyond the technology itself. So we are providing field support. We have over 25 field application scientists and sales. They work with our customers and partners really in a collaborative fashion, and that's, again, something that sort of continues to resonate through this meeting, I think. We have the master file, as I said, that provides some regulatory de-risking, so to speak. Again, the more times it's referenced, the broader the applications are, the sort of regulatory, the risk on the regulatory side is reduced, so to speak. And then just last focus in terms of partnerships is academic translational centers. So this has been a newer effort for MaxCyte, but a really big focus for us. And this really is to get in front of the science, get in front of some of the emerging strategies in the space. So we work with 25% of centers now to date, and we continue to build out this strategy. And again, this gives us it continues to build up our internal cell engineering expertise, which further helps on the partnership side. And I guess just the net here is we say we can deliver any molecule into any cell, and so we tried to put numbers around this. So this is our the inner circle is our clinical partners, and the outer circle is the market, so to speak. And so we really do work across the gamut in terms of cell type, strategic approach, indication. We've seen success moving beyond heme cancer into solid tumors. We support most, I think, if not all of the next generation sickle cell trials, for example, several of the allo CAR T trials. So I think this really sort of is sort of what I'd like to leave everybody with, and that the 20 years of experience we've developed does translate into diversity in terms of the partners we work with, and just in terms of our ability to support all sorts of next generation cell therapies. So I have 30 seconds left. I will leave it at that, and thanks again for having us. And again, there's plenty of MaxCyte people here in the audience and at the conference today for more questions. Thanks.